Dr. Tantawi has extensive research and practical experience in entomology. He's one of the few experts in flies and maggots, and now is a clinical expert in maggot therapy as well.
Thank you.
We're just about there.
An accidental but safe and effective use of Lucilia cuprina in maggot debridement therapy in Alexandria, Egypt. This work has been published in the Journal of Medical Entomology in the May 2010. The calophryd fly, Lucilia cuprina, is known to cause malign meiosis, and therefore it has been avoided in maggot therapy, whereas its sibling species, Lucilia sericata, breeds in carrion, and it is used in maggot therapy. The present study reports an accidental involvement of Lucilia cuprina in maggot debridement therapy in Alexandria that has proved to be safe and effective. Maggot therapy has been practiced in Alexandria since the year 2000 by fusing disinfected maggots from laboratory colonies descended from third instars collected on carcasses placed in the botanical garden of the Faculty of Science. Since 1988, all of the Lucilia larvae were collected by these methods, and I found that adults of Lucilia cuprina were extremely scarce and did not breed in carrion. In November 2008, we renewed the colonies by collecting third instars by the same way, and based on many years' experience that the species would be Lucilia sericata. As part of a quality assurance protocol, the new maggots were screened using molecular identification, and we discovered that the species is Lucilia cuprina and not Lucilia sericata. The new maggots have been successfully fused for maggot debridement therapy before being screened. And if so, I surveyed again the carrion fly community in the botanical garden, and I found that the two species coexist and breed in carrion. Therefore, the presence of Lucilia sericata in carrion in Alexandria and its use in maggot debridement therapy are new records. The molecular identification with this table shows the primer fused to amplify and sequence the CFO1, the 20FS, and the pair gene of the new maggots. The result here show that the Egyptian specimen showed if 90 to 100% match with the specimens of Lucilia cuprina in New Zealand and Australia. For the morphological identification, adults and larvae of the two species are extremely similar in appearance. The two species can only be separated under a microscope by a skilled taxonomist. Here, the two species can be separated by the number of CT on the posterior area of the head. So in Lucilia cuprina, there is only one theta under the inner vertical theta, whereas in Lucilia sericata there are about 3 to 4 theta. This is the lateral view of the thorax of the blowfly. The two species can be also separated by the number of theta present on the posterior border of the notopleuron and the humeral callus. So here, in Lucilia sericata, the number of CT, they are about 7, 8, but always in Lucilia sericata, they are only 3 or 2. The third instars of the two species, the larvae, the third instar, can be separated by the dorsal tubercles on the last abdominal segment. In sericata, the distance between the inner dorsal tubercles is equal to the inner and the adjacent median one. Whereas in cuprina, the distance between the inner dorsal tubercle is greater than the distance between the inner and the adjacent medial one. The effectiveness of Lucilia cuprina in maggot therapy, the larvae were successfully fused to completely heal the wounds of 2 diabetic patients. Each patient received 2 cycles of free fringe maggots. And I will comment only on one case because it is very important. The patient had 2 wounds on his left foot, one on the lateral side of the foot and the other wound on the plantar surface. The lateral wound was completely debrided by the first cycle of maggots, and we applied intentionally the second cycle on the clean wound to enhance healing. This is the clean wound before the second cycle, and this is after the second cycle, and this is after 1 week of the second cycle, after 3 weeks of the second cycle, here complete healing. So this proves that our strain of Lucilia cuprina is safe. The lessons learned and discussion. Any new population of a particular fly species must be taxonomically identified before establishing a colony of it in the lab, especially where sibling species coexist. And of course, we are aware that if the introduced species had been applied to the wounds of the patients and found to be invasive, the patient may have suffered. The primary role of the medical entomologist is to select a safe and an effective species or a strain to ensure the success and the safety of the treatment. Two possible explanations of the recent occurrence of Lucilia cuprina in carrion in Alexandria, maybe because of the human agency. The pupae of the species was associated with the introduced plants, and maybe due to the climate change. Therefore, the entomologist should verify the blowfly species composition in their local habitat on a regular basis, because it may change due to exotic species introduction or global climatic change. The present study also highlights the question of whether different strains of the same species can be more or invasive, safe or effective. There are certain strains of Lucilia sericata, the widely used species in maggot therapy, that have been known to invade the healthy tissue. And we explain this, that phthiasis has evolved independently in geographically isolated populations of the same species. Therefore, despite the safety of this strain of Lucilia cuprina in maggot debridement therapy, entomologists rearing blowflies for the purpose of wound debridement should regularly maintain high quality assurance of their species identity to avoid the possible clinical complications that may result from the introduction of an unexpected and invasive species to their laboratory colonies. Because the number of fly species, if used therapeutically, is limited at present, the potential use of more species should be encouraged. So in Alexandria, 3 blowfly species has been successfully fused: Lucilia sericata, Chrysomia albiceps, Lucilia cuprina. And we found that the maggots of Lucilia sericata were more effective in debridement than those of Chrysomia albiceps, perhaps due to a difference in the potency of the proteolytic enzymes of the two species. And as we know, that the second and third instars of Chrysomia albiceps are facultative predators on larvae of the same and other species, whereas if those of Lucilia sericata are pure scavengers throughout their entire feeding period. And this photo shows the third instar of Chrysomia albiceps devouring the third instar of Lucilia sericata. So if a Chrysomia albiceps, of which it differs somewhat from Lucilia sericata in its biology, can be used safely for maggot debridement therapy, why not we use Chrysomia, why not we use Lucilia cuprina? The last slide here, it is my own thought and ideas. Here is the decision of whether larvae feed on dead or living tissue maternally induced, which means that meiosis is a natural phenomenon in which the female blowfly experiences the wound before laying the eggs. In maggot debridement therapy, the female blowfly is allowed to lay the eggs on dead tissue. And we can see that in the United Kingdom, Lucilia sericata causes serious meiosis in sheep, but still used in maggot debridement therapy. To date, no invasive blowfly species has been recorded in a well-prepared maggot debridement therapy application. The primary screw-worm Cochlomia the primary screw-worm fly, Cochlomia homonivorax, which causes malign meiosis, can be freed in the laboratory on dead tissue. And this, the maggots of this species eats only healthy tissue in the nature. Okay, and maternal diapause is well known in the larvae of the blowflies and the pupae of the flesh flies in response to short day lengths experienced by the adult female fly. And thank you.